going to start the session now. So the first team is from Aberdeen in Scotland, and you've got 20 minutes. Okay? Thank you.
Good morning, everyone. Welcome to our presentation. We are the Aberdeen 2014 iGEM team. I would like to start by introducing you to our team. My name is Martina, and together with me on stage I've got Anna Maria and Konstantin, and the rest of our team sits in the audience. We've got Jamie, James and Joseph. The aim of our project is to stop the HAT. HAT stands for Human African Trypanosomiasis. It is a vector-borne parasitic disease caused by a Trypanosoma gambiense parasite and spread by a bite of an infected tsetse fly. It affects mainly rural populations of sub-Saharan Africa, where infrastructure is poorly developed. World Health Organization classifies HAT as one of 17 neglected tropical diseases. That means that there has been no significant scientific or technological advance made in the field in the last 50 years. Although when detected early, HAT is fully curable, the disease still claims more than 9,000 lives each year. So why is it so hard to detect HAT early? In its genome, Trypanosoma gambiense codes for more than 1,000 different variant surface glycoproteins. Those are present on the antigenic coat of a parasite and are what in our immune system raises antibodies against. Unfortunately, by the time that those antibodies have time to be made and target Trypanosoma for destruction, the parasite undergoes so-called antigenic shift, in which it degrades all of its antigens and replaces them with a different, brand new set of VSGs, which are no longer recognized by the antibodies. Therefore, the capability of the Trypanosoma parasite to change its antigenic shift so often renders it invisible to the immune system for use. There are only two different VSGs that are always present in all HAT sufferers in the early stages of the disease. Those are called LETAT 1.5 and 1.3. They are expressed early in the disease and cause a strong antigenic response, and antibodies made against those epitopes persist in blood long after antigenic shift occurs. What further complicates early diagnosis of HAT are unspecific symptoms in the early stages of the disease. Those mimic flu and therefore seem benign and are often neglected. It is only in the second stage of the disease when the true neurological symptoms of the disease become apparent. Those include poor coordination, disturbed sleeping patterns, as well as psychiatric disorders and coma, which, if untreated, always proves fatal. All current diagnostic systems look for the presence of Trypanosoma-specific antibodies in the blood sample of a suspected HAT sufferer. The most common test, card agglutination tests, is not specific or sensitive enough to allow detection alone due to cross-reactions, therefore needs to be followed by a risky procedure of a lumbar puncture, in which a sample of a cerebrospinal fluid is taken and visually investigated under the microscope for a presence of living parasites. In addition to low sensitivity and specificity, all current diagnostic methods require bulky equipment, as well as reagents that need to be stored under 4 degrees. Obviously, this is not ideal for a system that is supposed to be used in the field in Africa, where temperatures are more than 10 times higher. Therefore, our team started to wonder whether it is possible to engineer an alternative diagnostic system which would allow early detection of HAT, which would be tailor-made for use in sub-Saharan Africa. Our team brainstormed and came up with a list of features that we believe such a system should display in order to be a successful replacement for current diagnostic methods. We decided that sensitivity and specificity are of highest importance, since all current diagnostic systems lack those. In addition, since it will be used in tropical countries, we decided it needs to be thermostable, inexpensive, as well as portable. If we manage to engineer a system that is simple and gives a measurable output, we would make the diagnostic system easy to use and therefore eliminate the need for highly trained medical staff. We decided that synthetic biology offers tools that are tailor-made for engineering of such a device. In our system, we use E. coli surface display of rain epitopes, as well as an AND-gate logic, which we implemented with the use of quorum sensing. We started off by engineering 2 separate strains of E. coli, a sender strain and a receiver strain. Our sender strain produces homoserine lactone, whereas our receiver strain senses the presence of those in the surrounding and responds by a production of green fluorescent protein. Both strains express via different autotransporters different epitopes that mimic Trypanosoma-specific epitopes. Our system looks for the presence of two trypanosoma-specific antibodies in a patient sample, lit at 1.5 and 1.3. Therefore, there are four possible outcomes of our diagnostic test. In the first case, no trypanosoma-specific antibodies are detected, and therefore the outcome is negative. If we only have a single trypanosoma-specific antibody present, it is either sender or a receiver that bind. Since communication between those two strains is not possible, the outcome is still a negative. It is only in the last case, when both trypanosoma-specific antibodies are present, and therefore both sender and receiver bind, only their communication via homoserine lactones is possible, and therefore a production of GFP occurs and confirms a positive diagnosis of HAT.
So what did we actually do? We started by engineering autotransporters to allow cell surface display of those epitopes. We then tested quorum sensing by mathematical modeling and in the lab, and we finalized our project by creating a new and cheap GFP detector for HAT diagnosis. These autotransporter proteins are normally found in gram-negative bacteria. Within their coding region, they have all the necessary information to allow translocation of those foreign sequences on the cell surface. During our project, we focused on antigen 43 and nice nucleation protein. Both of them are composed of an alpha and beta domain, the beta domain being involved in creating a pore to the membrane through which the alpha domain and other foreign sequences can translocate themselves to the cell surface. We inserted what's known as a flag tag, which is a protein tag, just before codon 148 with antigen 43, and on the C-terminal with the ice nucleation protein. We started with antigen 43, which had 6 additional PST1 sites within its coding region. We removed those through site-directed mutagenesis, and then we inserted a flag sequence which had 2 restriction Cas sites, both in antigen 43 and in IMP. We confirmed this by sequencing. We then used the Western blot to confirm that we have flag within the cell lysate. This is IMP, this is antigen 43 full length, this is the alpha domain. And we just focused on antigen 43 to remove 2 beta herpins, which are known to be involved in cell-to-cell aggregation and biofilm formation, because we wanted to avoid any false positives that this would bring to our system. We confirmed by sequencing again, and we compare them to a negative control, which is AG43 only. You can see this one is aggregating, whereas this one is not anymore. We then will use what's known as the immunolabeling technique to see if flag is indeed on the cell surface. This is a negative control. These are E. coli cells which express AG43 only, therefore we can see no immunofluorescent E. coli cells. On the right-hand side, we have E. coli cells which express AG43 plus flag. You can see a ring-like structure surrounding E. coli cells, suggesting that yes, flag is expressed on the cell surface. After that, we focus on the actual assay. On the left-hand side, this is the ideal plan. We have 2 antibodies, we have quorum sensing happening, we have GFP output, therefore we can say that the person has been infected with HAT. However, we wondered, what happens if you have senders which are not bound to the antibody? Are they still able to quorum sense and still result in a GFP output significantly, meaning that we have a false positive? This is what mathematical modeling came across with our answers.
So in order to eliminate any false positives and make sure our system is working as we planned, we look at spatial modeling of our quorum sensing system. We wanted to use the diffusion equation, which is a partial differential equation, and we wanted to relate the production of HSL in time to its spread as a field in space. We also used the flux equation. This is a normal gas flux. For the people that don't know, this basically means that all the HSL that leaves the cell needs to travel through an imaginary surface around the cell. We could relate those 2 equations and come up with a model to simulate our system. If we look here at initial modeling, this is just as a single E. coli cell. It produces a field of concentration of HSL, and we can see that HSL diffuses so fast that about 2 or 3 cell radii, we can barely detect HSL. It's about less than 5% of its initial concentration. If we look here, where we simulate multiple cells, we wanted to see if aggregation of cells would have any local impact to quorum sensing. Again, our initial model confirms that even though cells here are aggregated, the quorum sensing molecule concentration field is still very low. It's not impacted by local concentration and aggregation of cells. Therefore, we concluded that quorum sensing is solely due to the background of HSL in the medium, and juxtaposed bacteria did not produce enhanced QS, quorum sensing. So how do we produce enhanced quorum sensing? Well, we looked at geometries. Our flux equation initially looks like that for a free-floating bacteria. But if we bind a bacteria to a surface, we can see that the area through which the HSL needs to travel is reduced. That means that the same HSL is produced, but now it travels to a smaller area, therefore the concentration is higher. We believe that that would increase our quorum sensing response and enhance the system. We confirmed that in an experiment where we have freely suspended cells and surface immobilized cells to a polylysin plate. We can see that after about 300 minutes, the surface immobilized cells gradually and steadily enhance quorum sensing and produce GFP to a much higher rate than our freely suspended cells, therefore confirming our model. So to summarize, this is our ideal project. We want this. We want cells to bind and communicate through quorum sensing. We want this to be enhanced, so that's why we stick them to a surface, but we don't want extra cells interfering. And that's why we need to wash. And I'll let Anna-Maria explain more about this.
Okay, so at this stage we know the answers from mathematical modeling. What we did further in the lab is to use a pull-down assay combined with quorum sensing to see if we can detect this happening in the lab. So, in figure 1, we have a negative control. In this one, you have receivers which do not express Ag43 plus flag. Therefore, when you incubate them together, they're not able to bind. You add the senders artificially, and therefore you have no GFP response. However, this one is our construct. This is what we wanted to test. You have receiver E. coli cells which express Ag43 plus flag. They can interact with the magnetic bead via the anti-flag antibody, and by the end of the assay, when you add your senders, you have GFP response. Well, did it work? Well, yes, it did. This one is our negative control. Once again, receiver plus sender, you have a linear GFP response. However, this is our construct. Therefore, you can see a definite increase in GFP, suggesting that yes, it worked. So at this stage, we wanted to focus on the actual assay. We have antigen 43 plus flag, and we have 2 restriction cassites. We can then replace that with what's known as mimotopes. Mimotopes are shorter versions of epitopes, known for the HAT-specific antibodies. They are better because they do not cause cross-reactions. How do you start? You start with a human blood sample taken from a person. You suspect them suffering from HAT. You immobilize the antibodies on a polylysin coat. You then add your pre-washed E. coli cell, which expresses mimotope VSG 1.3, and your sender plasmid. You then wash one time on PBS, because mathematical modeling told us before that yes, it's important to wash in order to remove, to remove unspecific binding. And then you add your pre-washed E. coli cells, which express the second mimotope, which is VSG 1.5, and your receiver plasmid. You wash again because it's important. You incubate them together, which allows them to communicate via HSL. And if by the end of the assay, you have a GFP output, you say that a person has been infected with HAT, unfortunately.
Yeah, but what good is GFP in Africa? Any of you can see GFP with your eyes? Well, in Africa, they don't have microscopes. And an industrial fluorimeter costs $1,000. So we wanted to solve this problem. We wanted to create a device that you can buy with a single banknote, that is portable, so you can take it in your bag. It's made for dummies, you don't need any training, and it's very robust. So we came up with this. We illuminate, we have a culture that we suspect has GFP in. We illuminate with a UV LED at a specific 395 nanometer wavelength. And on the other side, we get fluorescence green, some other diffracted colors, and leftover UV. We filter those with 2 filters, UV filter and a green Pasteur filter that only lets through green light. and we detect this with an LDR, this light-dependent resistor. We connect this to an Arduino, which is our analog to digital converter, and then we analyze the data in a Raspberry Pi. So did this work? Yes, it did. This is a device that holds all the components, and you can see here we've tested on our laptop connected directly. This is actual data from quorum sensing bacteria. So after about 100 and something minutes of incubation, we were able to detect quorum sensing and GFP in our culture.
So this is our proposed diagnostic backpack. As you can see, it comes with both our sender and receiver strings, as well as untransformed E. coli to remove any unspecific binding. We've got polyolysin-coated Eppendorf tubes, as well as saline buffer solution and filter papers to carry out the wash steps. Vacutainers allow easy withdrawal of blood. Our portable GFP device also easily fits there, and together with solar panels, forms a self-contained diagnostic system adapted for use in tropical countries. We believe that over the summer we engineered an alternative diagnostic system for HAT, which is highly specific due to the fact that a colocalization of both trypanosoma-specific antibodies is needed for a positive response, as well as the fact that we use mimotopes, which are more specific and do not cause cross-reactions. Since we operate our system in bacteria, it is thermostable as well as sensitive, since amplification of response via homoserine lactone is possible. Our device is inexpensive and basically can replenish as bacteria grow. It is highly portable and simple to use. The output given by our GFP device is very simple and does not need medical staff for analysis. Since iGEM is not only about lab work, we focused on a number of human practices. Some of them include interview at a local radio station in Aberdeen, as well as presentation at the Explorathon 2014. We will happily answer any of your questions during questions time regarding our human practices and outreach. At this stage, we would like to summarize our achievements over the summer. We successfully optimized autotransporters for display of foreign peptide sequences on the cell surface of E. coli. We optimized our assay with mathematical modeling, which guided our actions in the lab throughout the summer. We successfully demonstrated empirically that quorum sensing behavior is a viable option for detection of colocalized disease epitopes. Last but not least, we built a portable GFP detector for less than $100, therefore delivering a proof of concept that synthetic biology can be used to diagnose neglected tropical diseases. Overall, we believe that the best aspect of our assay is that it is a platform technology and can be easily adapted to diagnose any neglected tropical disease you might wish. At this stage, we would like to thank our sponsors and supporters, without whom our work this summer would not be possible, in particular our supervisors. Thank you very much for your attention, and we welcome your questions now.
Questions from the judges first.
I have a question regarding the biobricks. If I look at the registry, there's little information or no information. Can you still enter this, or why did you not enter any information?
Do you mean any biobricks in particular? Because we did submit all of our entries.
What did you showed some data here, but the data is not on the registry.
For the biobricks.
Should be all
Yeah,
registered.
we did submit all of our entries.
Okay, you submitted them, or did you enter the information in the registry?
We did enter information in the registry.
But at least the parts I have here.
What parts in particular are you looking at just now?
Okay, we can discuss this.
Yeah.
So first of all, I applaud your efforts in doing things safely and in accordance with the different review boards that you had to go through to get this project approved. You mentioned when you approached your, to get approval to work on genetic material that you had to put into your application, anything you think you might ever need. And you commented on your wiki that that might slow progress because you can't anticipate beforehand where the science is going to take you. So I'm just wondering if you had any thoughts on how that review process might be different so that it wouldn't hinder the science.
I think the review process itself is aimed at providing a level of security to everyone who's aware of it. In some ways, although the review process, we've said we're only restricted certain things, that can work out in the favour sometimes. A lot of our discussions with the public revolved around the views of genetic modification of organisms and whether or not it was right and whether it should be made more available. And the general consensus was that strict review processes should be kept in place to increase public confidence in the science. So while it would hinder the science itself, I think it would probably be best to keep things the way they are for the time being.
Yeah, and I agree with you. And I agree that that's what came across in your wiki, but you said it might slow down. Is there some way to change the review process, which I agree is necessary, that to help include the science? So no, that is a good answer. That's a good answer.
Yeah, very nice work done here. I'm interested to know the time course and the detection time. Can you show us the slide? Because the initial time seems to be quite long. Was that 300 minutes? For the GFP to come out and then the machine to detect? Oh yeah, okay, 100 minutes. But how many times have you repeated this testing? And also, have you tested with other standard colors or frozen materials?
Yes, so the way this works is our assay works as we mix the two cultures, we wash, and then we let them to grow and quorum sense. So quorum sensing takes time. But if you see on our previous slides, where we actually show lab data, you can see here that even a professional fluorescent microscope can only detect quorum sensing after 150 minutes of incubation time. So our very cheap device doing it in 100 minutes is still very impressive.
There is a latent period for the quorum sensing,
yes.
Regardless of the test. It will be around 300 minutes, and that's when you'll be able to tell there's a difference.
So how quantitative would this device be in detecting the parasite? How quantitative that would be? Have you tested with any other standard?
Okay, so we haven't actually done a sensitivity analysis. We've done a proof of concept. We've shown it can work. But of course, full sensitivity analysis with a live disease is going to take much more time than this.
So, just turning to your AND gate. AND gates are notoriously difficult to stabilize in terms of biological systems. Can you just say more about your AND gate design, how you balance the inputs to get stability? And also, did you actually do this in the wet lab, or was this just a theoretical exercise?
We did do this in the wet lab, and we kind of want to take the biology out of the AND gate. So if we go back, so what we basically do is the false AND gate could happen if we have extra centers that are not bound anywhere. But that's why we wash, so we kind of eliminate the biological unreliability there. So we do this mechanically by washing twice after we add each material. And that's how we make sure that our AND gate works. And we did a successful experiment. I think Anna explained, this one, right? And yeah, this confirms our AND gate.
I very much like the synthetic biology in your project. Can you also explain the advantages versus a normal ELISA, which would have the antigen immobilized and then detect the antibody by a secondary antibody?
So the main issue with that is that in Africa, you don't have access to refrigeration which is good enough for storing antibodies. So they may be stored at -80 or -20. Periodically, you've got cool boxes which keep it at 4 degrees, but then, well, they're gone. They get destroyed and they're expensive. So this negates the whole problem of having to use antibodies in the first place to use the patients.
Can I just quickly add to that? The other advantage, of course, is that with the biological system, you don't need to keep going back to the manufacturer to get fresh supplies. The nature of a bacteria is you just need, antibiotic, agar. You can keep replenishing your supplies without ever needing to go back to the manufacturer.
Quick question on the temperature effects. Have you tested the effects of temperature on your device and all the reactions? Since you have not mentioned what temperature have you performed the experiments and the testing?
Well, this is all live bacteria that we're doing it, and it's been incubated at 37 degrees Celsius. We haven't put it on maybe the degrees that it would be in Africa and the humidity, but there are no moving parts. The device is fully reversed, so I'm guessing that wouldn't affect at all because there's no mechanical.
Yeah, so E. coli can survive temperatures up to 37, they don't happily grow in that. You can also use this if you wish in the bacillus, which can form spores, that's 5 up to 60 degrees.
Okay, we need to draw the questioning to a close now. Thank you very much. So the next team can come up.
I was like, no, I didn't even question.
What is the code for our library?